understand from. Yeah, we go. I will revise again that one. Musis. Yes. Now I will again show you what you need to remember. Significance of meiosis. Meiosis make gametes. What are gametes? Gametes are male sperm cell, female ovum or ova. Okay, last part. Genetic, why it is important to make the genetic variation. Then we divide it is into a meiosis 1 and meiosis 2. Right? Meiosis 1, you remember what is happening at prophase 1 and metaphase 1. This one, right? This you need to remember. And metaphase 1, random assortment. Again, important these two for genetic variation. Now after that, anaphase 1, the homologous chromosome separate. Homologous chromosome separate. Telophase 1, again, it form the cells. Then coming to the next stage, prophase 2, metaphase 2, anaphase 2, telophase 2. This is same as mitosis. Same as mitosis. Events are same as mitosis. So prophase 1, what is the 5 things you remembered? It happened here. And metaphase 2, the only one you remember that the chromosomes align at the equator. Same thing apply. Anaphase 2, chromosomes separate from the centromere because of the pulling of the microtubules. Check it. Mitosis, it is happening. And telophase 2, again, 2 cells are formed. Then again, starting a nuclear membrane. All things are same as uh, telophase of mitosis. Then the common event is cytokinesis for any event, the division of cytoplasm. Again, filaments, microfilaments, make a string around it to make the cleavage furrow and then separate them. Don't make complicated. Certain lessons, you should avoid the distracting parts because some lessons, because of other information, it get distracted. So we should also wise enough, capable enough to remove unnecessary distracting part and only filter important things to remember. Right? So because there's another thing like it's same for anyone. Like when we study, the beginning it's complicated. Any lesson, physics, chemistry, biology, you see, that's complicated. If you feel complicated, you can segment it. Chemistry, physics, biology, whatever, segment it. What is segment? First, try to understand the very basic things, like the easy thing that you can understand, easy thing to remember. Then leave it. Then again take for study. Then you little bit add on to that. Likewise, when you add one by one layer, I think at one point it become easy that you remember everything, you understand well. Any lesson, if you see complicated, don't try to understand everything at the same time. So you just segment. Okay, now this I understand well. I can remember this well. Fine. I leave it. After 2 days or next week, I will again come and try to see this one. The next week, you can add a little bit onto that, right? And also, I should tell you, uh, for teaching purpose, this kind of repetition learning and something called spiral repetition spaces like things also existing. Don't teach everything at one time for certain lessons. Like we teach something here and add the complex thing later on. So it's common for many lessons, even medical faculties. They don't teach everything at the first lesson. They teach one, and maybe another lesson they add on to that. Like it's like a repeating again. Then you understand well. Okay, I learned this one, and again I'm learning. And addition to that, this is I'm going to learn. So methods are important. Keep in mind also that one. Okay. Now the last part, we're going to finish almost. We finished almost this mitosis. Um, Cancer, tumor. These words are very common. We increasingly hearing this word, cancer and tumor, like words, right? Uh, the cancer is a dangerous condition because at the moment many deaths are due to cancer. If you look WHO ranking of the diseases with non-infectious diseases, I mean not infectious. Uh, Infectious mean it's caused by microorganism. Others are called non-infectious. It is not infectious or non-communicable. That means other person will not get. Highest death all over the world rank with cardiovascular disease. Heart and blood vessel related disease is number one. After that, you can see rest of the list. I think on the top first few of these top cancer also listed because uh, if I don't want to get diabetes, if I don't want to get heart attack, people say, okay, avoid these risk factors. Example, heart diseases. Consider your food, not too much fat, not too much carbohydrate. Don't inactive, do exercise. Quit smoking. Reduce stress, reduce salt like things given. But cancer, how a person can avoid cancer? At the moment, it's very difficult because we don't know actually what are the all the risk factors for cancer. It's getting higher at the moment, but our aim is not to learn about the cancer as a lesson here, right? Now, this word, tumor, and this word cancer, at A levels, it is not that strict exact meaning of this word, but there is a proper meaning for this word. So, meaning is, let's say tumor first. Now, if there's a cell and this cell lost its cell cycle control, it lost cell cycle control. Why it is lost cell cycle control? The regulator of the cell cycle are not properly working. It is not important for you to understand and keep this knowledge, but I will tell you additional things because if it is something interested, And if you don't tell it, I mean, it's knowledge. That is the knowledge we need. Like when you're doing A level, you need to read not the Campbell and resource book. There are many other interesting articles, findings, so many things. You need to read these things because for interest, right? Now, what we know at the basic level, as I told you before, the cell cycle is controlled by the DNA because there's no other molecule. So far, DNA is the molecule. So, what is inside the DNA? Genes. So, DNA molecules divided into a special sequence order called genes. What genes make? Genes make proteins. DNA make proteins. And what are the proteins? Okay, here in this case, there are two group of proteins. One is called cyclin, other one is cyclin dependent kinases. Right? So these are the two players. They need to activate each other, and the cyclin and cyclin dependent kinases, they can control the cell cycle. So as we discuss checkpoints, G1, S, G2 level, they can do some things, whether it's going forward or not. Now, what will happen if something externally or internally can alter this gene, right? Let's see a chemical or maybe harmful rays, exposure to harmful rays, exposure to chemicals. So they're going to change the chemical structure of this DNA, especially these genes. There are a set of genes you don't want to remember, you don't want to learn. Tumor suppressor gene, proto-oncogenes, and within that again a special gene called P58 family genes. So these are the genes. They are there to make this cyclin and cyclin dependent kinases. They control the cell cycle. If this gene is altered, you can understand what is altered, right? Altered mean nucleotide order change. We will learn under mutation what is this and how it exactly happening. We call it mutation, change of the DNA sequence, some DNA nucleotide. It is called mutation. If this happens, the protein is altered. It's not the correct proteins. If this is altered, these players, cyclin and cyclin dependent, cannot control cell cycle. So what will happen now? Uh, the cells start to divide. Cell divide in an uncontrolled way, like this, which is not the expected one. We never expected this kind of a growth of cells, and now the cell mass, it form a cell mass. So tumor is considered as a increased number of cells to make a visible structure. The cell number increase to make a visible structure, and it is stay there. No harm. There's a growth, and it is stay there. But when it comes to cancer, this become not. This is not anymore innocent. It's growing not only that place. It is growing many part of the body. That means it spread many part of the body. If there is a growth, let's say you see a person, he has a growth inside the neck. That is the increased cell number, and it's you see a big growth inside this neck area. But cancer is one spreading all over the body, right? So it can spread, then it become a cancer. Right? So we use this word. Uh, okay, here a man who lived with a tumor on his neck for nearly a quarter of a century has finally had it removed. I mean, quarter of century. So you can see how long he lived, like a 25, yeah, 25 years. This is the growth, and called tumor. The word is tumor is used. So it's he can live. He's not dying. It's the appearance is not good, and maybe it's a little bit uncomfortable with the person. But he lived 25 years. Maybe he thought to remove it at the end. So this kind of a growth, if we observed, it is called tumor. But uh, we don't know this tumor is tumor or tumor become a cancer. Right? So another interesting thing, uh, do you know that the human body has 37, 30 trillion cells? The human body has 30 trillion cells. Now, we can understand if the cell number increase, cell should divide. When the cell divide, there could be some errors happen, mistake happen. If this mistake make a mutation and mutation happen in this kind of genes, it turn up to a cancer. But you know, elephants they have 1,000 trillion cells. Elephant has this much of cell. Then who has more chance to get cancer? Elephants, because their cell division is higher than human. More cells divide. More divide, errors can happen. They can get cancer. But surprisingly, they don't get cancer. And scientists found they have extra copies of gene that code for protein P53. So the genes which control cell cycle, and you see here which has strong cancer preventing properties. What does it mean? It controls cell cycle. So if the cell cycle is well controlled and more backup genes are there to control, then less chance of getting cancer. I think one thing we can also learn, if our genes strong, we have a good genes, we have a powerful cancer preventing genes, it is better that person is getting less cancer, no chance of getting cancer. Finally, whatever we can take medicine, vaccine, or other things, if our genes strong, we are strong, right? Right. Then, when it into a, if it is not spreading, we call benign tumor. Not spreading and remain at the same place. So we use this word benign. Mostly, it is not harmful. Person can remove it. There's no chance of spreading, and it's called benign tumor. So people should not panic always when they have a growth start to grow some places in the body, observing something, but it's alarming because person cannot decide whether it's a tumor situation or cancer situation. Only has to see a doctor to find whether it's a cancerous growth or benign growth. So if it is cancer, the cancer has a special property. That means if this is the cancer, there is a chance, because these all cells are cancerous cells, right? It says usually start with one cell. So it is logical, right? It is logical for us because one cell divide by mitosis to make two cells. Two cell become four cell, eight cells. So then it can increase in number exponentially, and at the end, it all the cells are cancerous. Now the danger is if this cell can spread. Spread mean, in our body there are two transport system. One is blood vessels, and the other one is called lymph, blood and lymph. If these cells detach from the original site, and if it enter to this place, there's a chance they can travel to another place. So it's not that easy. That means I draw one cell is going out, and the cell can go to another place and make cancer. But it's not that easy, because it's not happening easily, because we have a protection system. So the body is trying to protect from harmful things. But if this cell is powerful enough, maybe it's going to another place and they start to grow. This is called a malignant, and this process is called metastasis. Spreading of cancer from one place to another place is called metastasis, and this cancer is called malignant cancer. So they can spread to vital organs like liver, kidney, and brain, or any other place. With the time, the person experience organ failure and widespread changes in the body. So this can end up with definitely death. It's very hard to detect if it is internal cancer and it is spread all over the body. So you can see here a breast cancer, which is common cancer, and how it looks. Here, the growth is like this. You can see small mass. They are growing like this. So if it is inside the body, very difficult to judge, because it doesn't make a pain, it doesn't make a fever, it doesn't make anything until it's spread all over the body. It's very hard to cure. Right, the last thing, metastasis is the spreading of a cancer cell to another place and start to grow. And the malignant is the cancer which has this capacity to spread all over the body. Malignant cancer, benign tumor, we call like this, right? Same thing we can observe in plants. Again, there is a abnormal growth in certain places. This is because of mostly the insect or other organisms. They stimulate cells to grow at certain place. So probably some insect they secrete saliva. Saliva stimulate the cells to grow, because you can learn two hormones, auxin, cytokinin. These two hormones important for the cell division of the plants. So this hormone actually controls cell division and the growth. So they can coordinate, but unnecessary stimulation of these hormones by insect, or maybe small worms or any parasites, they form this abnormal growth called galls. This is just in the resource book has a two last lesson. I think most important part is the meiosis and mitosis, right? Why is that? We should also understand at the end of the lesson uh, where is the area the question can be asked. So we had to always look in the past papers what is the area examiners would like to ask questions, because the examiners try to find questions in a relevant area, then they make question from that important area. Then we have to pay more attention in our lessons where is the most important things. Always we have to be confident 100%. Right, take your time and also check your notes if you have. Which of the following statement regarding eukaryotic cell cycle is correct? You can uh, check your notes, check time, whether this week. I know that after end of lesson you can't remember everything. So you need to check. It's like exercise, like check the notes. Okay, this area I learned cell cycle, DNA replication I learned here whether it's correct. Like when you, it's like a if you understand, if you remember everything, it is fine. So this is something important to get an idea what kind of a question asked and what are the areas asked, because the answer at the moment your answer is not that important. It is just exercise for you to look a little bit here and there and get an idea. It's okay wrong or right answer, you can answer well if you study everything, because your brain has all information. You can quickly bring every information, recall it, you can get the answer. But at the end of the lesson you can't remember everything, so you have to look sometimes with the notes like this. Okay, I just highlight few things before we find. Okay, you all answered. Right, starting from propase, I told you to remember five changes. This is mitosis, propase. There are five changes. Three propase, two at end of propase, that is prometapase. So also I explained what is the difference between chromatin, chromatid, chromosome. There are three different words: chromatin, chromatid, chromosome. Now, chromatin is the one usually we see in the nucleus. At any given time, if we look nucleus, we can see chromatin. What are chromatin? DNA plus proteins. DNA plus proteins. So inside the nucleus, we can see chromatin. They arrange, wrap around the proteins, and they stay inside the nucleus as chromatin. There's no formation of chromatin during the cell cycle or any place. They naturally they stay there as chromatin. Now DNA replicate. After replication, it make a copy. It is called chromatid, and DNA condense to make chromosome. Chromatin is DNA plus proteins, and if they divide, the sister chromosome is called chromatid. So this X-shaped structure is called chromosome because DNA is compressed and also duplicated. So chromosome, chromatin, uh, chromosome, chromatid, and chromatin. Many times I highlighted this, right? Okay, then the changes: chromosome visible, nucleolus disappear, spindle form. Third one is spindle form. Spindle form at propase, right? Spindle form at propase. And the fourth and fifth one at prometapase, chromosome more visible, chromosome attached to kinetochore. Nucleonuclear disappear, chromosome attached to kinetochore. So that's the five changes, right? Chromosome begins to condense, nucleolus begin to disappear, spindle form. Nucleonuclear disintegrate, chromosome attached to kinetochore. Okay, now let's go to the question. So these two hormones, I think we have to learn in the future in the plant's structure and function. But you hear it now. Okay, auxin and cytokinin. There is two hormones which is responsible for cell growth and cell division. If you hear it next time again like this, it's going to your head, right? Okay, very strongly. We need to remember the crossing over. We learn synapses, synaptomental complex form, crossing over, chiasma. So this all happen in prophase 1, right? This happen in the prophase 1. It's not metaphase. Prophase 1 of meiosis 1. Prophase 1 of meiosis 1. Then it's correct. So here it's wrong. It's happening metaphase, right? Formation of chromatin. Now this word is chromatin. Chromatin is the one usually we can see in the cells because chromatin is DNA plus proteins. There is no need to formation, only chromosomes forms. The DNA replicate is one event, chromatid form, chromosome form, right? Not chromatin. DNA replication occurs in S phase, not G2 phase. Nuclear envelope reform during cytokinesis. Now, nuclear envelope reforms happen in telophase. So, at the end of telophase, start to make nuclear envelope. So, nuclear envelope form, nucleolus form, chromosome visibility is gone. If you look prophase 5 changes, spindle form at the prophase. Spindle form at the prophase. Now, what you have to learn from this question, exam is targeting mitosis and meiosis, the events happening at each stage. That is the most important target for the questions, right? So you should exercise to remember the events of all the stages. It takes time, but after few weeks and few months, I think you remember all events because within 1 to 1 and a right? Yeah, 1 minute, let's say. So the lesson we look in few hours, like two classes, few hours. You have to recall all your knowledge for within a minute to bring these changes. So it is not that easy. Only it is possible if you remember everything very quickly. So